Okay, I'm going to give a brief review of the activity from Chapter 2. So this is for students that might not have understand the answers to these problems or just wanted a little bit more information on the logic behind why I designed the activity the way I did. So this key is now available on the Canvas website as a PDF, and it is the answers to this activity. So here we are asked to consider an organism with a diploid number of 4. And you're also told that the size of the genome is 20 megabases of sequence. So this is a pretty small genome. Humans have 2 billion, so 2,000 megabases, so 2 gigabases of sequence. But there actually is a large variation in genome size. So this is actually a possible range for the genome size. And it's also actually a possible range for the number of chromosomes. So the first thing that you needed to do was remember that the diploid number tells you how many unique chromosomes there are. And so if the diploid number is 4, then if you just solve for this equation, 2n, then we know that the number of unique chromosomes is 2. And so there's a chromosome 1 and there's a chromosome 2, and then each of those has 2 copies, 2 homologous chromosomes, one from the mother and one from the father. And so I asked you to both draw out the chromosome structures at various stages of mitosis and meiosis, and I also asked you to think about things like the base pairs of DNA, the number of chromosomes, and the number of DNA molecules in the cell at these times. And hopefully this forces you to really think through what actually makes up a chromosome and think through how many DNA molecules are actually in the cell at any given time. So the first thing that the enterprising student might notice is that I asked you to look at the beginning of interphase and end of interphase, but these are not actually stages of mitosis. These are actually stages of the cell cycle. And at these stages, if you draw the DNA within the cell, it's important to note that the DNA is not organized at this time. It's much more randomly distributed throughout the nucleus. So we have not drawn a nucleus here, but you should also remember that DNA is contained in a nucleus within a eukaryotic cell. What happens, what is the key difference between the beginning and end of interphase? So you should also know this is the synthesis of DNA. So DNA replication has occurred, and so each strand has been replicated. And so the way we've depicted this is we've just copy and pasted this DNA to show that there's now twice as much DNA for each DNA molecule. Prophase occurs. And the P for prophase I always think of as preparatory phase, so this is when condensation actually starts to happen. And so the key step in prophase is that chromosomes form, these structures that now you should recognize as X shapes form, and that's because the DNA is actively condensed into these structures. And so how many structures do you draw? This is where the diploid number again becomes important. You simply draw you simply draw the diploid number of chromosomes. So if this was a human cell, the diploid number would be 46, and you would see 46 chromosomes within a cell at this point. So the way we've chosen to draw these is we distinguish the homologous chromosomes by color, and we distinguish the unique chromosomes by size. And this is pretty standard. So chromosome 1 from the mother is depicted in red, and it's the largest chromosome. This is in another standard that typically is followed. You typically number the chromosomes by their size, and then you also have a chromosome 1 from the father, which we've drawn in blue. Chromosome 2 from the mother in red, chromosome 2 from the father in blue. The key difference between prophase and metaphase is that these line up at the midplane. So this is in preparation of dividing these into 2 cells, and so the way that happens is that you have spindle poles and microtubules that form in between these and connect to the actual chromosomes. Anaphase, this is the active phase, so this is when the DNA is actually being separated into the 2 cells, and so now we see the sister chromatids have actually separated from each other, and eventually the cell will divide at this point right here. And then finally at the end of telophase, we've divided the cell. One cell has become 2, and so each one has a conservative set of chromosomes as what was starting out. So what that means is that the DNA content of these 2 cells is identical to each other. Both of them have a chromosome 1 from the father, chromosome 1 from the mother, chromosome 2 from the father and a chromosome 2 from the mother. So now let's look at number of chromosomes. We'll start out there just because I think it's probably easier. So the first two stages, this is a bit of a trick question. So in interphase, because the chromosomes haven't actually condensed, then I would say the most correct answer is undefined. So when we refer to a chromosome, typically what we're referring to is a physical structure, something that you can see under the microscope. And so those don't actually exist during interphase. At the end of prophase, there's 4 of them, metaphase 4, and then anaphase. We've split these actually into 2. So each chromosome has now been split. The proteins that formerly held these DNA molecules together are actually degraded. The spindle poles and the microtubules and the molecules that actually provide the force are now splitting and moving the DNA from the midplane into the new cell. And so we say that the chromosomes have doubled from 4 to 8 because of that. And then finally at the end of telophase, because we separated, there's now 4 per cell. The number of DNA molecules, so each one of these is a DNA molecule, and the end of telophase is essentially the same DNA content as the beginning of interphase. So there's 4 DNA molecules, and then at the end of interphase there's 8. And again, that's because DNA synthesis has occurred, and so we've doubled the number of DNA molecules. The number of DNA molecules in prophase is 8, so note that it's double the number of chromosomes. And that's because each chromosome actually consists of 2 DNA molecules. So there's 2 sister chromatids. Each sister chromatid is a single DNA molecule, and then these are exact copies of each other. So these are the copies that occurred because of DNA replication. Nothing changes in metaphase, and then at the end of anaphase we note that even though the chromosome number has doubled, the DNA molecules has not doubled. So this split just goes from 2 DNA molecules per chromosome to 1 DNA molecule per chromosome. And then finally we have it because at the end of telophase we know that the DNA content is half per cell. The base pairs of DNA, so we're told that there's 20 megabases of sequence in the genome, and so you should immediately think that's how much DNA is within a haploid cell. That's how much DNA would be in a sperm cell or an oocyte. What we're looking at the beginning of interphase, though, is a diploid cell, and so you have to double that number to get 40 megabases. DNA synthesis occurs, and so now we have to double it again because every DNA strand is duplicated. And so we go from 40 megabases to 80 megabases of DNA. And then again, nothing changes until we reach telophase, and we've halved the DNA content. Meiosis, what happens there? So here we are depicting the end of prophase 1. So these numbers are all the same, 80 megabases of DNA, 4 chromosomes, 8 DNA molecules, because meiosis and mitosis essentially start from the same point. So DNA replication has occurred. And the important thing to draw during prophase 1 is that the tetrad has formed. So the homologous chromosomes have actually found each other, and so these are also physically connected to each other through protein contacts. And so this is for the purpose of recombination. We haven't drawn recombination here, but if we wanted to be extremely accurate, we would also show that there would be exchange between the paternal and maternal chromosomes. End of metaphase 1. These have aligned at the midplane, and so again, the way that they're going to go is to the left and right of the cell. And so the goal of meiosis 1, you should always remember, is to separate the homologous chromosomes. And so here we've shown that at the end of telophase, that each of these cells now only contains 1 homologous chromosome. So this one has the maternal chromosome 1 and the paternal chromosome 2, and then the opposite is true for the other cell. And so we've halved the base pair, so 80 to 40. We've also halved the number of chromosomes because we've divided them, the chromosomes, into different cells. So the splitting hasn't actually occurred. So another thing to remember is that even though there's physical contacts between the tetrads, we don't say that this is the tetrad is a single chromosome. There's 2 chromosomes in a tetrad. It's confusing, a little bit confusing, I know, but it's the way it is. And then the DNA molecules half as well. And now metaphase 2, the goal of meiosis 2, you remember, is to split up the sister chromatids. And so we align those at the midplane, again going left and right. Notice the arrows did not exactly work, but hopefully you get the point that they are going in the opposite directions. And then at the end of telophase 2, we now have the DNA content. So we're back to the genome content, so 20 megabases is the same as the genome content, so that looks good. And that's because we've halved the DNA by going from 2 cells to 4 cells. The number of chromosomes doesn't change because we split each one and then halved it by cytokinesis. And then the number of DNA molecules is now down to 2. So we have a chromosome 1 from the mother, chromosome 2 from the father. This one is the same. This one has a chromosome 1 from the father and a chromosome 2 from the mother, and then these 2 cells are the same. So these cells would not be the same if recombination has occurred, but when we ignore recombination, we have 2 identical cells and then 2 more identical cells. But the important thing to note that this is not a conservative process, so the DNA contents of these 4 cells is not the same. So this is distinguished from mitosis, where the DNA content is the same, and it's the same as the starting cell. At the end of meiosis, we have different DNA contents for the different cells. So that's the extent of what I wanted to cover, and hopefully this activity helped you understand the Chapter 2 material.